As I mentioned at the beginning of class, the idea of this is, what happens if you have genes that are on the same chromosome, and especially if they're really close to each other on the same chromosome? How does that affect, when we are looking at probabilities of how different offspring can be inherited? So that idea is called linked genes. So a linked gene is, again, genes that are located in the same chromosome, and they tend to be inherited together because they're not going to get split up whenever you're doing independent assortment. Or we're going to talk about when we do crossing over, they don't get separated as often during crossing over. Okay. The idea of this would be, it kind of reminds me of like you're ever shuffling a deck of cards. If you have maybe cards that are really, really close together, sometimes they tend to travel together in the deck of cards, right? You got to keep shuffling it over and over multiple times. Be kind of the idea. Each chromosome got hundreds to thousands of genes, except the Y chromosome for males, so it's pretty small. All right, moving on. This is kind of where you're getting into with the fruit fly lab a little bit later, where a lot of our discovery of some of the genetics of linkage came from what you're going to be doing in the virtual lab, and what Morgan did in his actual lab was breeding fruit flies and carefully observing the movement of traits across generations, kind of like what Mendel did, but having the benefit of some modern genetics. Okay. And the two traits that he focused on especially were body color and wing size. So he tried to pay attention to how did those traits, body color and wing size, how do they get passed on? And so what he found when he carefully observed some of the numbers was that body color and wing size, they typically are inherited together. They typically would travel together. And what that essentially made him conclude was those two genes that control those traits, they must be really close together on the same chromosome, since they don't separate that often. So there you go. And here would be the idea of it. So let's say you have two organisms bring together. You have a dihybrid female. So B plus would be referring to what they call the wild type. I'll show. I'm gonna have a slide where I show you what the flies look like. But anyways, a female who's heterozygous for the two traits, and then a male who's homozygous for the two traits. And most of the offspring, when they come together, they're going to look like this, where this chromosome here will maybe pair up with that one, or that chromosome there will pair up with that one. That's what you would expect. Now, we now know that you have crossing over, and so you'll get more combinations. But initially, what Morgan thought was you would get either this or that. That's what you would typically think. And just a reminder, it's a test cross is whenever you're crossing somebody who's recessive for both traits. So here, just lowercase b with no plus sign, that would be considered recessive. And again, I'll explain what the traits look like here in a minute. Anyways, but when he did this, he realized that you would get non-parental phenotypes produced. In other words, not all the offspring looked like this or like this. And so that led to the question of, well, why? Because it should be either you get this or you get that, right? If you do a Punnett square, the Punnett square should look like one of those combinations or one of those combinations. So looking at it, that's where we get into this idea of genetic recombination, which comes about by crossing over. Okay, so looking at kind of the cross with it. So let me write kind of the key for some of these traits. So they're looking at if you were B plus, this would be considered dominant. And B plus would go with a gray body, gray, and they call this the wild type. Basically, the wild type is compared to the non-mutant type. So you'll notice here they say mutant. It's like the non-change type. What the flies typically will look like. And then if you are just a lowercase b, that would be the mutant phenotype, or the non-wild type. So non-wild. And what that actually looks like here would be black body. So you'd have a black body. and that would be considered recessive here. Okay. And then the second trait that we're looking at would be for the wing shape, where the wild type would be VG plus, and that means you just have normal wings. As in just like your garden variety fly kind of wings, right? The wings are what a fly would normally want. Whereas the mutant wings would be VG without a plus sign, and those would be, they call them vestigial wings. Basically what that means is like, vestigial is kind of referring to like a remnant of something. Like the wings are like a remnant, like the wings are stunted in growth, okay? That would be a mutant, again, that'd be a mutant type, because that's not what you would normally want. So normal wings would be considered the wild type, the vestigial wings would be considered the mutant type here. All right, so in our parent generation, you took one fly that is homozygous for both the dominant traits, all right? So they're B plus, VG plus, VG plus. And you're gonna cross it with a double mutant. So somebody who has all of the recessive alleles. So essentially you're doing like a test cross. And then the first female you would get here, so following that arrow, would be a hybrid called a dihybrid. They'd be heterozygous for both those traits. So then what we're going to do is we're going to do a test cross. We're going to cross her with another double mutant, another fly that's got all lowercase b's and they're all lower, b's without the plus signs and the VGs without the plus signs. Now looking at potential offspring here, the female, the eggs she could create would be essentially all of the 4 possible kinds of eggs, right? Now, the reason why this dihybrid cross doesn't look as big as some of the other ones is because this male can only make one kind of sperm. He can only make BVG without any plus signs, okay? So these would be your 4 possible offsprings. You'd have a wild type, that would just be getting your garden variety fly. One that would be a double mutant, so black and vestigial. One that would be a partial mutant, so it would have the normal wild type gray body, but then it would have the vestigial wings, so the mutant wings. And then one that would be a mutant black color, but then would have the normal wild type wings. All right, so going from there, so this picture was too big. So here's the same, this is where we ended on the last slide. Now, what you would expect if these genes, because what we're looking at is, are these genes on the same chromosome? If they were on different chromosomes, what you would expect is you would get an equal number of all of these offspring. Equal chance you get this, equal chance you get that, or that. Now, if they're located on the same chromosome, what you would expect is you would only be able to get this offspring or this offspring. It would not be possible to get either of these offspring. Now, what Morgan found was neither of those two things happened. What he got was most of the offspring were looking like one of these first two, but some of them were the ones where like they end up having like the heterozygous. It ended up being a hybrid for the 2 traits. So this is where it gets into this idea of linkage and crossing over. So how it so the basis of recombination, again, that's the crossing over that was happening during prophase 1 of meiosis. A little bit of the vocabulary. The offspring that the non-parental phenotypes, what they mean by non-parental phenotypes are these 2 flies, the ones that were hybrid for the 2 traits. We call those recombinant types or recombinants because, again, they had a phenotype that was not one of the parents. This last bullet point, I'll explain a little on that, like kind of going through some of the math on the next slide. But essentially, a 50% frequency of recombination, if they're on 2 different genes, would be saying that you would have, that's just saying this right here, that you would have equal chance of getting either of those offsprings. But I'll show you the math on why it's 50%. Okay, so let's say if we're looking at unlinked genes, which when Mendel did his pea plants, the genes for pea plants, they were unlinked, meaning the genes for the pea shape, so wrinkle versus round, or color, yellow versus green, those genes were on different chromosomes. They were unlinked, meaning you had the same chance of getting this versus that, right? It'd be 1 to 1 to 1 to 1. Okay, equal chance of getting either of those offspring, assuming you crossed like a parent who was heterozygous for both traits, and then you did a test cross, you crossed it with a parent that was homozygous recessive for all of those traits. You would get 1 to 1 to 1. All right, so what Morgan found, again, was that he didn't get 1 to 1 to 1 to 1. He ended up getting some offspring that had a mixture that were hybrids for that. Okay, that's where Morgan came up with this idea that these there must be some sort of crossing over, some sort of exchanging of those genes. And if you're a little confused right now, you should be. Let me show you a picture, and it'll start to like kind of come together. Hopefully, here's what's going on. So we take our wild type female here. This is what the wild type female's chromosomes would look like, right? So she's got, B plus, VG plus, B plus, VG plus. Those are 2 chromosomes. Then you got that double mutant male, and he is recessive for both of those. Okay. When these 2 come together and have an offspring, they made that one dihybrid female, where she had 1 copy of the B plus, VG plus, 1 copy of B, VG. Okay. And if we did that test cross where we then crossed her with a double mutant male who's, BVG, BVG, same genetics as this one up here, these are the different possible, like they would then go through S phase of meiosis. So they're going to make a second copy. So what would this be called? What are those called? We did S phase. Now you get that second chromosome coming in there. What do we call those? Sister chromatids, right? So you got your sisters here. So now you got the formation of the sister chromatids. Okay, so then continuing this picture. So here's the 2 chromosomes. You done S phase. You have the sister chromatids present now. So now during the first meiosis, we had crossing over. So this is how this is why Morgan got the different numbers of his like offspring, because you have crossing over happens. You now have the ability for an organism to have to pass on to their sperm or their eggs a chromosome that is a mixture. That's the wild type and the mutant type, you would say, plus and then no plus. Okay, that's the idea. Because crossing over happens, you're not getting either the wild type or the mutant type. So moving on with it. And this is showing the males going over. Now I have a question for you. That male here that was like a double mutant, how come they're not showing crossing over happening for this male? Does that mean the males like these chromosomes don't do crossing over? They do crossing over, but why don't they bother showing that here? Actually, really important you understand that, because if you don't if you can't see that, I don't think you're really understanding what is actually happening here. Why are they not copying these chromosomes down here and showing crossing over happening? Because they're not, you don't have to show crossing over because they have the same DNA. If you cross over these chromosomes, you would end up with the exact same chromosomes. Crossing over is only relevant when it's 2 different, you had 2 different copies of the genes from the parents. You see that? Because now you can get a pink portion and a red portion, pink portion, red portion. If you do crossing over here, you would just get 2 of the same blue chromosomes. That's why crossing over isn't relevant if you're either homozygous dominant or homozygous recessive. Yeah, go ahead.
So crossing over occurs both chromosomes within a pair.
Can crossing over occur with both chromosomes in a pair?
Yeah.
Are you saying like could this chromosome cross over with that chromosome?
Yeah.
No, it would only occur within this one pairing here. So crossing over is only occurring between these 2 chromosomes. Crossing over only occurs with the sister chromatids. Would be like the proper way to say it. Only the sister chromatids are doing the crossing over. That's it. Yeah.
If they're both like heterozygous, crossing over still occur.
If they're both heterozygous, does crossing over still occur? Yeah, and that's what you're seeing here, right? Because they're this individual is heterozygous for this trait, that is why crossing over is giving you a bunch of different combinations. Yeah, that's key. Whereas if you're non-heterozygous, crossing over doesn't really do much for you for those two genes. Okay, so moving on. These are the possible eggs that female could make. She could make an egg that would be her normal wild type. She can make two eggs that are, she can make one egg that's a wild type for both genes. She can make one egg that would be a mutant egg for both traits. Or she can make two recombinant chromosomes. We call it recombinant because they get a mixture of one wild type and one mutant gene. Okay, two of those. Now, if these, and then these are the offspring that they came from. So they mated 2,300 offspring. So Morgan had 2,300 fly babies. If these chromosomes were on, if these genes were on different chromosomes, you should get an equal number of every offspring. He should have gotten, it would be 575 of these, 575 of these, 575 of these, 575 of these. That's what the results would be if two genes you're comparing are on different chromosomes, if they are unlinked. Okay? Now, you know genes are linked, and you can determine how linked they are, how close they are on the same chromosome, by the number of recombinant offspring you get. The more recombinant offspring you get, the more unlinked they are, because the better the chances are that they're going to cross over. The fewer recombinant offspring you get, the closer they are on the chromosome, because it's rarer that they're going to cross over. Again, think of you're shuffling a deck of cards. The closer two cards are to each other, the more likely it is that those two cards are going to stay closer together after you've finished shuffling the deck of cards. The further the cards are, the more likely chance they're going to remain further, would be kind of the idea. So how the math works is, what he did was 2,300 total offspring. Of those 2,300 total offspring, 391 were recombinants. They were one of the hybrid for both traits. Where the 391 came from was 206 plus 185. Okay, so what that tells, and then you times it by 100, and you get a 17% linkage. So what that means math-wise is, we call it the map distance. That 17% would be the map distance, the distance those two genes are from each other. In other words, if I draw like if I go back to this slide, if I draw like a basic chromosome here, or I'll just use their chromosome here. What that 17% is saying is these two genes, you would say they're apart from each other by 17%. Now, let's say if you did the same thing, you would analyze these two different traits, and you found that you had like a recombinance that were, say, like 38% for two different genes. What that would tell you is that the two genes would be like here and, say, here. They would be further apart on the same chromosome. And then if you got a 50%, now understand how you get 50%. You would get 50% if 50% would happen if out of the 2,300 total offspring, 50% would happen, I said, if you had 575. So if these genes were unlinked, you would get equal numbers of both offspring. So when you did the math, if you do 575 plus 575, the total number of recombinants would be 1,150. 575 plus 575? Yeah, 1,150 divided by 2,300 times 100, that would give you 50%. What that would mean would be that those two genes you analyze are on two different chromosomes. Okay? I'm going to do another sample problem on that, but it's a goofy idea. I promise you it's not as complicated as maybe you might be thinking right now. I know it's kind of, might seem like a lot. It's a simple idea, but the point is, the idea is the closer you are to 50%, the more unlinked chromosomes are, the smaller the percentage you get, meaning the fewer recombinants you get, that means they're closer together, the two genes. All right, I'll keep moving on with it. Here's a slide just kind of showing you the basic formula. So this is the formula that I was using, where the map distance, the distance two genes are from each other on a chromosome, are the number of recombinant products. So on that last slide, that would be the 206 and the 185, divided by the total number of products, the total number of offspring. And that last example is 2,300. And if you did that math, we got 17%. And if your percentage is less than 50%, then that's going to tell you that those genes are linked. If you get 50%, that would tell you those genes are completely unlinked. So in other words, linked and unlinked, there's a spectrum for it. Okay, some genes maybe like slightly linked. Maybe that means they're very far apart on the same chromosome. They also will use the unit called map unit. So sometimes you'll see this as like, for this one, 50%. This would be the same thing as 50 map units. So percentage and map units just go 1 to 1. This last bullet point, I'll explain when I show you another practice problem with it. So let's do one other problem, and this would be kind of the example of a problem you might see like on the AP exam or something. So they're telling you got 2 plants and they have 100 offspring. 5 of those offspring are red and short, 10 are white and tall, 40 are red and tall, and 45 are white and short. And they're telling you that red and short, so red and short and white and tall, those are the recombinants. They came about by recombination. So if they say calculate the recombinant frequency, RF is recombinant frequency, to figure out are those genes linked? Are the genes for height and color, are those 2 genes linked? So what you would do is you would use this formula where you would have the total number of recombinant products on top. So what would be my total number of recombinant products? In other words, how many red and short, or red and short plus, let's call red and short RS. How many red and short and how many white and tall, we'll call it WT, do we have in this problem? How many do we make? Red and short, we have 5. So yeah, 15. 5 red and short, and white and tall are 10. Okay, so 5 plus 10 would be 15. Now the total number of offspring is what? 100. Okay, good. And then you're going to multiply it by 100 to get into a percentage. So 5 plus 10 is 15. Divided by 100 times 100, those just cancel. So you get 15%. So what that tells you is there is a 15% linkage here. 15% is less than 50%. So what does that tell you? That's a good question. This is key. Yeah, it's less than 50%. What did I say 50% meant? They're unlinked. They're on different chromosomes. They're crossing over and separating completely independent of each other. So if you're less than 50%, what does that tell you?
Yeah.
They're linked. Good. That's the math you got to do. The math is not that complicated, but you got to understand 50%. Again, where the 50% comes from is if you had equal numbers of all 4 of these, half of them, 50% of them would be recombinants, 50% of them would look like the parents. That's why 50% is the marker you're looking for. Okay, I want to do, and then I have that written out for you. And I drew out here like what it looks like on, like if you were to draw like a hypothetical chromosome. So the idea would be you have the color gene and the height gene, and you would say they are 15% or 15 map units, you could say, apart from each other. The higher, the more recombinants you get, the further apart those genes would get. Joshua? Yeah.
So are recombinances
Recombinants mean they look different from the parents. So like recombinants here, the parents, the 2 parent flies that we started with, one was the wild type for both. They looked gray and had normal wings. And then the other parent phenotype was the double mutant, so it looked black and had like the janky wings. So then recombinants, those are the ones that don't look like the parents.
So how does this how is that different
These would be recessive. It's related to recessive, but it isn't exactly the same as recessive because it's all about like what did your parents start off with? If that makes sense. Yeah. Anyways, I got one more problem for you. It's a little bit different. And this is one where they'd be less likely to ask you about this. I'll just show you what the picture looks like. So let's say they give you, all right, they're telling you they got they got a gene pair gene B and GCN. There's a 9% recombination frequency between them. 9% is that's pretty low. What that tells you is that these two genes are linked. B and CN, they're very close to each other on the chromosome because the 9% is low. CN and VG is 9.5%. So CN and VG, the distance between them is 9.5%. So then the total from B to VG would be 17%, they're telling you. So the idea would be, could you draw a map for these? And by drawing that, it's literally just like, put it, if you have B to CN is this, CN to VG is that, and B to VG is this, you just kind of line them up with each other. And you can keep doing this until you can make a complete map of all of like the fly genes. And that's what like, you know, the different scientists that worked with fruit flies, that's just sort of what they did for their careers. Just like carefully doing some of these calculations and tracking these traits to create a map of where the genes are at. Nowadays, you could actually like do like DNA sequencing to figure this out. But this is how they did it back in the day when they didn't have actual DNA sequencing to rely on. Now, you may be wondering why 17%? Like, you're like, 9 plus 9.5 is not 17%. What? It's like, and here, you're right, it's not. What's going on here is whenever crossing over would happen, B to CN could swap. But then you could have VG and CN could then swap. So you go back to your starting place. So the reason why it's 17%, not 18.5%, is because of that. Like you could end up it's like if you shuffle a deck of cards and like say you shuffled it once and like you switch two cards, then you shuffle again and you put them back, two cards back in the same position they were before you ever started shuffling the cards. That's what's going on here. That's kind of a technicality. That's kind of confusing. It's not a big deal, but I just wanted to point out the obvious of why that's not 18.5%. So just because sometimes you undo each other when you do the crossing over. Okay. All right. Last thing is just pointing out how recombination relates to evolution. Remember, a key concept of evolution is evolution needs variation. If all organisms look the same, there's nothing for evolution to work on. No one organism can be favored over another. But if you do recombination and again random fertilization, random sperm can fertilize random eggs, then that's when you can get even more combinations, more variation. More variation, there's more for natural selection to select upon. Okay, let me stop the recording.